All right, so I'm going to pick up with linkage, and I did skip a couple of slides, so please make sure you review the PowerPoint or outline because I'm not making a recording on the couple of slides that I missed. So we've been talking about sex-linked traits, which were traits that were linked or carried on the X chromosome, but lots of traits are linked, and any two traits that are carried on the same chromosome are called linked traits or linked genes. And really, your chromosomes have thousands of genes on them, so it's not surprising that once in a while you might be studying a trait or two traits, and it turns out that those two traits are carried on the same chromosome. And if they are, and they're linked genes, they're not going to go through independent assortment. Instead, they are going to get stuck together and be inherited sort of as a package deal. Now, that probably doesn't make a whole lot of sense. I'm going to show it to you in a picture. So let me switch over and let me show you this. So here's what we're saying. So if you have genes, if you have an individual that's big A, big B, little b, we have been assuming that A and B are on two separate chromosomes. So notice here, one set of chromosomes carries A, and then a different homologous pair carries B. And that's why we had to come up with all these possibilities that you could get big A with big B, big A with little b, little a with big B, little. That was called independent assortment because it's completely random which copy of A ends up with which copy of B. Now, here's the thing though. If the genes are on the same chromosome, you would still be big A, big B, little b, but now the genes are what we call linked. Well, as you can imagine, since the chromosomes are what go through independent assortment, this entire chromosome is going to go one direction, and this entire chromosome is going to go another direction, which means the only two possible outcomes now are big A with big B and little a with little b. And so that is what is going to change our outcome if two genes are linked. You're not going to see the possibilities of big A with little b, little a with big B, the ones I've highlighted on the left here. Those two sperm or egg combinations don't exist. So going back here, the test that we do to see if two genes are linked is called a dihybrid test cross. So we're crossing one that's heterozygous for both of the traits we're looking at with one that is homozygous recessive, just like in a regular test cross. Now, here's the outcome we would expect to see. I actually drew this out here. Oh, wrong one. Here. Okay. So if the genes are not linked, you would solve this on the left just like we did in class. Now, I did it with the product rule, but a dihybrid test cross, so if I cross a tall black with a short white, my expected outcome is 1/4 tall black, 1/4 short white, 1/4 short black, 1/4 short white. The ones that look like the parents, what do I mean look like the parents? That have the same, remember, one of my parents was tall and black, one of my parents was short and white. So the offspring that are tall and black or short and white are called parentals because they look like the parents. The other two combinations, short and black and tall and white, are called recombinants because they have a new combination. In other words, neither of the parents was short and black. So this is a new combination of the two genes. So if the genes are not linked, and they are on separate chromosomes, like my picture here on the left, the genes are on separate chromosomes, we would do the cross just like we did in the previous chapter, and we would expect 1/4 approximately of each of the possibilities, and about half or 50% would look like the parents and 50% would not. Now on the right, I've done the same cross, but if the genes are linked. So if the genes are linked, notice what happens here. A and B are on one chromosome, capital A and capital B. Little a and little b are on the other chromosome, and I actually solved this one with a Punnett square because the only two possibilities here now are capital A with capital B, little a with little b. Notice that what we get is that all the offspring would look like one of the two parents. Half would be tall and black, half would be short and white, and notice that we don't see any with these recombinant short black and tall white. Those don't exist. So that's the difference. So application-wise, if you were to see a cross on a test and they gave you the outcome, they showed you the offspring, if you saw approximately 1/4 of the offspring of each type, it would mean that the genes are not linked, that you're seeing regular independent assortment, the genes are on separate chromosomes. If most of the offspring look like one of the parents, mostly parentals with very few recombinants, it means the genes are linked. Now you might say, well, I don't understand. Why would you get any recombinants at all? So let me show you this next slide. So this is what they're showing. So this trait, the wild type flies are gray bodies, and normal wings. So gray bodies with normal wings. That's the regular fly. That's called wild type. It's the one we see in the wild, sorry. Gray, normal. And the other combination is black flies with vestigial wings. So they're using big B for gray, and they're doing this. So big B, little b, big, they're using VG for vestigial, big, little here. So big, little, and they're crossing it with little, little. So this is the dihybrid test cross. And what they're trying to show, I know it's probably a little confusing, this parent can only give little b, little VG, but this parent can give 4 possibilities. They can give big, B, all these 4 combinations. And this is like a Punnett square. So this is little b and little VG. And basically, if you look here, they got, in the outcome, it's sort of cut off, but they got 484 offspring that were black and vestigial, 465 that were wild type. They got 472 gray and vestigial, and 490 that were black and normal. The parentals are the wild type and the black vestigial. These are your parentals because they look like the parents. These are our recombinants. Notice that we get about 1/4 of each, and about half look like the parents, and about half look like recombinants. Now, what if they're linked? Well, if they're linked, big B and VG are on one chromosome, little b and little VG are on the other. So now the number of possibilities is much lower. You would expect to just get black vestigial and wild type and none of the other 2, but yet they did get some. And again, the numbers got cut off, so I'm going to rewrite them. They got 944 of these, 965 of these, 206 of these, and 185 of these. And the question is, why did we get any of these? And what they're trying to show you here is it's because of crossing over. Remember in the previous chapter, we talked about how genes can cross over between homologous chromosomes. So you're not going to get all parentals, but you're going to get mostly parentals. And one of the things you could be asked to do would be to actually calculate the percentage of parentals and the percentage of recombinants. So the percentage of parentals is going to be the number of offspring that actually look like the parents over the total times 100. Now, if the genes are not linked, that should be about 50%. And your recombinants is going to be the number with the new combination that's not in one of the parents over the total times 100. And if they're not linked, that should also equal 50%. If they're linked, if the genes are on the same chromosome, what we're going to see is this is going to be much, much, much higher, probably 80% or higher, and this is going to be 20% or lower. Now, the last thing I want to tell you is when they calculate these percentages of recombinants, we can use that information to tell us how close together genes are on a chromosome. So in other words, let's say they provide you with this data. So this is recombination frequency data. So what they're saying is 18%, when they do a cross and look at genes A and B, about 18% of the time we see recombinants. In B and C it's 5%, and in A and C it's 13%. The rule is the higher the percentage, the farther apart the genes are on the chromosome. So they could ask you, they could give you these pictures, and these are showing sort of a map of the 3 genes and their locations on a supposed chromosome. And they could ask you which of these 3 maps properly maps out the locations of these 3 genes. And the correct answer would be number 3. Why? Because if you notice, A and B have the highest percentage of recombination. That means they have to be the farthest apart on the chromosome. And notice in this picture, A and B are the farthest apart. Now, if you did have 2 answer choices and both had A and B farthest apart, then you could also look at the fact that B and C should be the closest together because their percent is the lowest. In this scenario, in my 3 choices, only one of the 3 choices actually had A and B the farthest apart. But it is possible on the AP exam you could have more than one choice. So your first step, find the 2 that are farthest apart, that's your highest percent, and then work from there. Cross out everything else. If you still have two answers that might work, look at your other percentages. The lower the percent, the closer together they should be. The higher the percent, the farther apart they should be. And that's going to be your clue as to which one is which. So, wrapping this up, again, why do we see some offspring that are recombinants if the genes are linked when you would expect none? Because of crossing over. And the higher the crossover percent, and I have this down here, the higher the crossover frequency, the farther apart they are on the chromosome. Bottom line, if you don't understand all of that work that I did, which would be very unfortunate, but if you see a test question, when you do a dihybrid test cross, the expected is going to be 1/4 of each combination. If you see mostly parentals, and you've got to know what a parental is, that most of the offspring look like the parents, and very few recombinants, it's going to tell you the genes are linked. It's linkage. So usually what you're going to see is you're going to see a problem. They're going to show you a Punnett square. They're going to show you how many offspring of each type were found. Look at the offspring. If the offspring are about 1/4 of each type, you're going to say, oh, this is independent assortment, meaning that the genes are not linked, they're on separate chromosomes, and they are independently separating from each other. They're randomly separating, that's why you're getting that result. If you see almost all parents, it's going to tell you the genes are linked. You could also be asked to calculate the percentage of recombinants or the percentage of parentals. This picture is just showing what I was saying, that the farther apart the genes are, the higher the crossover frequency. So V, G, and B are the furthest apart, and then C, N was found in the middle. And notice the crossover frequencies are lower on these two, and it's 20 on this one. Again, I'll put this up one more time because I'm not sure if it's in the outline, but you definitely are responsible for it. You need to be able to calculate the percentage of parentals and the percentage of recombinants. And then look at those percentages. That's also going to tell you if genes are likely linked. High percentage of parentals is going to indicate they're not linked. They're on separate chromosomes. High percentage of recombinants, they are likely linked. And that is everything on linkage.